So, we will just do a recap of the assignment process in the case of nucleic acids, and by and large we do this by the nosy experiment, which we discussed last time in greater detail. And here is just to show that once more, this is a 2-dimensional nosy spectrum of a particular oligonucleotide whose sequence is given here on the top. So, this has 3 thymines here and are very well characterized by the presence of the methyl groups in the base. So, here you have a methyl group in the thymine base, and that is very distinctly seen at the top here. So, that actually fixes the positions of the thymine residues. So, and as we know, this one is the area which corresponds to the base protons, and the base protons, the NOEs are there to the sugar rings. These are the H1 prime protons of the sugar ring, and then you have these are to the H3 primes, and these are to the H4 primes, 5 primes, and then these are the 2 prime to double prime areas from the base protons H8 and H6 to the base protons, that is indicated here H8, H6, H2, but the NOEs come from the H8 and H6. You do not see so much from the H2 protons. All of these are base protons, and then you have here very characteristically these are the thymines. So, therefore, these are the NOEs from the methyls of the thymines to their own H6 protons. There are 3 of them here, you can see them very strongly here, these 3, and therefore this fixes the positions of the thymines here. So, everything what you do should be consistent with that. That actually provides a very good starting point here, because you see I will illustrate this to you in this particular sequence here. We saw this in the last time, but I will go over it again. You start with the T at the 5 prime end, then the T at the 3 prime end. So, what we have seen earlier was that you will see from the base proton NOEs to the sugar protons of the same nucleotide and to the sugar protons of the previous nucleotide, that is at the 5 prime end. So, therefore, this T does not have anyone on the top there. Okay, therefore, this thymine base will have only one connectivity from its base proton. Secondly, we also know where are the thymines. The thymine positions are fixed here. In this particular sequence there are 2 thymines, and the NOEs from the methyls to the base proton fix the 2 thymines to be here. So, therefore, one of the thymine is here, other thymine is here. Now, if you look at these 2 thymines, one of them shows a sequential connectivity here to the 5 prime end. Therefore, obviously this must correspond to this, whereas this one does not show any sequential peak. Therefore, this must correspond to this. Now, you can start from either end of these ones and you can walk, and ultimately you have to reach this place. Therefore, you can see from T8 self you see to the next one G7, from G7 you go to its own base, from the G7 you go to the G6, this is the sequential peak, then it will go to its own base, then it is G6 here, G6 you go to its the sequential one, you go to G5, then from G5 you go to the C4. Okay, now that is here, there is a sequential, this is the self. From C4 you go to the G3, and then that is the sequential connection here to this center here. Now you notice that this peak consists of 2 peaks here. Okay, so there is one sequential which is connecting, the self peak is here, and its sequential is also in this area, and this connects to the G2 here. From G2 you go to T1 and you reach here. Therefore, you see the sequential walk is easily done in this entire sequence. Once you have these 1 primes identified, you will also identify the 1 prime, 2 prime, the 2 prime, 2 double prime, and the 2 prime, 3 prime peaks from the NOEs which are occurring in this area. So, you have this 1 prime are identified here, you see NOEs to the 2 primes and the 2 double primes of the individual nucleotides, and also to the 3 primes here, and then to the 4 primes and the 5 primes here. Therefore, once the 1 primes are identified, you connect them to the 2 prime and 2 double prime. These are also seen in the cozy spectrum. 1 prime, 2 prime peaks will also be seen in the cozy spectrum. Therefore, once you have the 1 prime identified, you will have the 2 prime, 2 double primes identified there. Okay, you will also see the 2 prime, 2 double prime cross peaks here, and these we are clearly distinctly seen at a different place. And then you will have from the 1 prime you will have the 3 prime 4 prime peaks which are present here in this area. So, the whole spectrum therefore can be analyzed by once you get these 1 prime assignments here, and you also have this base to base assignments here in this area. Okay. So, having done these assignments, now you have the full assignments of the entire nucleotide. Now you have to go to the structure. The structure, as we said, consists of the backbone structure and then the sugar ring structure. The sugar ring we have seen earlier is called the sugar puckering. It is described by the pseudo rotation angle P, and we have all the sugar puckers are classified into two categories, north and the south, which we described earlier. The north corresponds to the C3 prime endogeometry, which is the center of the north region, and this happens in the A type DNA helix or in the RNAs, and in the B type double helix you have the C2 prime endogeometry. These are characteristically shown here. How do they look like? The sugar pucker is in this way. And then you have the glycosidic torsion angle here, which determines how the base is oriented with respect to the sugar ring there. Okay. So now we have to determine the sugar geometry. There is only one variable here, that is the pseudo rotation angle. And how do we determine the sugar geometry? What are our observables from the NMR point of view? And that is, the sugar ring, as you know, has many protons, and all these protons are coupled, J coupled. So you have this 1 prime on every position, you have the protons here, 1 prime, 2 prime, 2 double prime, then you have the 3 prime, then the 4 prime, and the 5 prime, it goes like that. So far as the sugar ring is concerned, we have these couplings which are present here, 1 bond, 3 bond couplings, and those ones are indicated here. So you will have the ribose ring, we will have these coupling constants here. I will show you the coupling constants a little bit more detail, and this is once again the pseudo rotation circle. So you have the southern part, this is the so called C2 prime endo region, and this is the northern part, this is the C3 prime endo region, all other intermediate puckers which occur in this area. Now how do we characterize these sugar rings? What are our observables? The observables are the coupling constants. The coupling constants are indicated here. Now the coupling constants depend on the sugar geometry. What is plotted here? How many coupling constants are there? We have 1 prime to 2 double prime, then 1 prime to 2 prime, 2 prime to 3 prime, 2 double prime to 3 prime, and 3 prime to 4 prime. So we have 5 different coupling constants. 5 different coupling constants which can be used to characterize the sugar ring. And these ones depend on the conformation of the sugar ring, and that you can see from here. These are plotted here, the coupling constants vary with P in this manner. Okay, this big one here, this is the 1 prime 2 prime coupling. This peaks here at this about 10 Hertz is the coupling constant here for the C2 prime endogeometry. Okay, for the C2 prime endogeometry, this 1 prime 2 prime coupling is 10 Hertz. So, and we can write here some of these things. So for the C2 prime endo, for the C2 prime endogeometry, you have the 1 prime 2 prime is approximately 10 Hertz, and you can look at the other ones, the 1 prime 2 double prime, 1 prime 2 double prime is approximately, you can see which one is that. That is the one which is below there. So that is approximately 6 Hertz. That is here. So in this area, this is 10 Hertz, this is 6 Hertz, and this one is my 2 prime 3 prime. What is 2 prime 3 prime? This is the third one, and that is approximately 7 Hertz. Okay, so that is about 7 to 8 Hertz. 7 to 8 Hertz. And what about 2 double prime 3 prime? 2 double prime 3 prime is this one. Okay, we are talking here. So this is about 2 Hertz or less than 2 Hertz, 1 to 2 Hertz. And 3 prime 4 prime 0. Okay, and these are very characteristic coupling constants, and they will show up in the cross-peak fine structures. In this we are going to discuss. Now what happens in the case of C3 prime endo? Let me write here the C3 prime endogeometry. So you can see what are the coupling constants. C3 prime endo means we are here. So here 1 prime 2 prime is 0. Okay, and all others are relatively larger, all others are greater than others, all others greater than 6 Hertz. In this area you can see this one is very small, this is the C3 prime endo area, this is 6 Hertz and this is about 7 Hertz, then you have 10 Hertz. Among these the strongest ones are the 3 prime 4 prime, this is the 3 prime 4 prime, this is the highest value for 10 Hertz, and then the 2 double prime 3 prime is also around 8 to 9 hertz. So that is this one here. So therefore these are very distinct coupling constants which will allow you to distinguish between the sugar geometries. Okay. Now how do they show up in the sugar geometry? So we will have to calculate the sugar geometry fine structure for this. Therefore we will try and calculate that in the COSY, we will have to use the COSY spectrum. Okay. So fine structures in the cross peaks of the sugar ring. First of all, let me draw the schematic of the COSY spectrum. What all you are going to get? So let us say I have this is the C3 prime endo. Let us say this is my, where are the 1 primes? Let us say this is the H1 prime, then I have 2 prime, 2 double prime here, then I have the 3 prime here, then these are the 4 primes, 5 prime, 5 double prime, 5 prime so we will not go. So up to the 4 prime one can go. So what do you expect here? From the 1 prime this is the diagonal, okay. So you will have the 1 prime to 2 prime to double prime. So you will, let us say this is my, I will draw that here, 2 prime to double prime, there are 2 here, then I will have the 3 prime and this is the 4 prime here, and then I have the 1 prime here. So this is the characteristic. Now what cross peaks I will have? Of course the diagonal will be present here. 1 prime to 2 prime to double prime peaks I will have 2 peaks here schematically in the COSY spectrum. So 2 prime to double prime. Of course you will have the cross peak between these 2 prime to double prime, there this will be present. Now 1 prime 2 prime peak, will it be present in the C3 prime endo geometry? For a general thing all these peaks will be there, but will this 1 prime 2 prime will be present, will it be present? So let us look at that. So 1 prime 2 prime coupling is 0, right. So for the C3 prime endo geometry this peak will be absent, okay. 2 prime double prime will be there. Now all other peaks will be there. Now 2 prime to 3 prime peak will be there though. So 2 prime to 3 prime peak will be there, okay, and 3 prime to 4 prime, that is here, 3 prime to 4 prime will be there, this is also very strong peak, okay. So on the other side I am not writing, these symmetrically they will also be present here, so I am not writing on the other side. So these peaks will be present, you will have 1 prime to 2 double prime and 2 prime to 3 prime, this is 2 prime to 2 double prime to 3 prime, will it be present? 2 double prime to 3 prime will also be present, and that will also be present here. 2 double prime to 3 prime, and 3 prime 4 prime, will it be present? Yes, that will be present, and that is this is 3 prime to 4 prime, this is the diagonal of the 3 prime, this is the diagonal of the 4 prime, and these are the diagonals of the 2 prime and 2 double prime and 2 primes there, okay. So these many peaks, these 4 cross peaks will be present in the case of a C3 prime endo geometry. Now let us do that for the 2 prime endo geometry. It is right here, C2 prime endo geometry. Now let me do the same thing here. You have the 2 prime, 2 double prime, then you have the 4 prime, then 3 prime, then the 1 prime. So let us draw the same thing here, 2 prime, 2 double prime, 4 prime, 3 prime and 1 prime. Now what peaks will be present here for the C2 prime endo geometry? Go back and look here, we wrote those ones here. What are the coupling constants? 1 prime 2 prime coupling is very large, therefore I will see the 1 prime 2 prime cross peak, okay. What I will not see? What I will not see is I am indicating below, the 3 prime 4 prime cross peak will not be seen because that coupling constant is 0. See, this coupling constant is 0. 3 prime 4 0, and the 2 double prime 3 prime is also very small, 1 to 2 hertz, therefore you may not see this one also. So what you will see? You will see these 3 couplings, these 3 couplings only will be seen, okay. So let us draw that here. So therefore, so I will have 1 prime 2 prime coupling, this peak will be present, 1 prime 2 double prime also will be present, and what about 2 prime 3 prime? 2 prime 3 prime will be present, 2 double prime 3 prime will not be present, and 3 prime 4 prime also will not be present. So therefore I will have only 3 peaks here on both sides. Of course correspondingly there will be symmetrically they will be present here as well. I am not writing on this side. So basically on one side these are distinctively different kinds of patterns you will get in the two kinds of a sugar geometry. By looking at this kind of a peaks itself you will be able to figure out whether I have the 3 prime endo geometry or the C prime endo geometry which is pure. In the case of the pure geometries. But of course if there are equilibrium mixtures of the two, then you may find the peaks present for the other ones also. So this we can figure out by actually measuring the coupling constants. Now how do we measure the coupling constants? The fine structures contain this information. All of these peaks they have fine structures, and the fine structures will have the information about the coupling constants. So how do we measure these coupling constants? In order to do that we must be able to understand what are the fine structures in these peaks. Okay. So let us take a particular peak, let us say cross peak 1 prime 2 double prime. Let me consider one particular peak 1 prime 2 double prime for a C2 prime endo geometry, for C2 prime endo geometry. So we have seen this how to calculate these fine structures, but let us do that once more here. So let us say you have the H1 prime chemical shift here, this is the H1 prime chemical shift, this is the H1 prime, this is the single line, this is the chemical shift here. What are the coupling constants this will have? This will have H1 prime has 1 prime 2 prime and 1 prime 2 double prime. These two couplings are there. Now if you are looking at the 1 prime 2 double prime cross peak, now I will have an active coupling and a passive coupling. Which is the active coupling here? Because I am looking at the 1 prime 2 double prime cross peak. Therefore this is my active coupling and this is my passive coupling. What is the consequence of this? The active coupling will lead to plus minus splitting. So I will split this here, 1 plus minus, this is the 1 prime 2 double prime coupling. Okay. So now we notice here in this 1 prime 2 double prime coupling is of the order of 6 to 7 hertz. Now 1 prime 2 prime coupling is much larger, that is about 10 hertz. So therefore this will further split into 2 and they will go like this. But this, since is a passive coupling, I will have this plus and plus here and minus there. So this will produce me a structure plus minus plus minus. Okay. Let us look at the 2 primes, the 2 double prime peak. Now if I draw the 2 double prime peak here, H2 double prime, what are the coupling constants for this? It has a 2 prime 2 double prime coupling, 2 prime 1 prime coupling and 2 double prime 3 prime coupling. Now this one has 3 couplings. Okay. So this one has 3 couplings, it has 2 double prime, 2 prime, 2 double prime, 1 prime and 2 double prime 3 prime, but 2 double prime 3 prime is very small. So okay, so 2 double prime 3 prime is almost close to 0, so therefore we will not see that, right. So 2 double prime has 2 double prime 1 prime coupling, 2 double prime 3 prime coupling which is small for the C2 prime endo geometry, and the 2 prime 2 double prime, this is a large one. This is typically of the order of 14 hertz, this is the geminal coupling, and this is of the order of 7 hertz. Okay. Now therefore what is the kind of a splitting here? So now let us draw this pattern here. So what is the active coupling? The active coupling is this one, this is the active coupling, right. So therefore if I want to draw first the splitting due to the active coupling, this will be plus minus and this will be of the order of 2, this is 2 double prime 1 prime coupling, and this will be further split into 2 because of the 2 prime 2 double prime coupling. Therefore this will be plus plus minus minus, this is 2 double prime 2 prime coupling, 2 double prime 2 prime coupling. So this also will be plus minus plus minus. So what will be the fine structure? So therefore I will have here plus minus, and here also I will have plus minus. So if I multiply this, how many components I will have? I will have 16 components. This peak will have 16 components. Therefore, I want to write that here inside plus minus, then I will have minus plus, this will be plus minus, plus. See, 16 components, this is the kind of a structure that will be present in the 1' to double prime peak. So if you look at the 1', 2' peak, of course then you will have to consider differently. In the case of 1', 2' peak, what are the, they have like 2', 3' coupling also in addition, okay. So go to the 1', 2' coupling, the same geometry, C2' endo. Now I want to see what will be for the 1', 2' peak, 1', 2'. See the 1', 2', now what are the actual coupling here? Let us split that here. 1', 2' coupling is very large, 1', 2' coupling, this is the active coupling now. 1' to double prime coupling will become smaller, that is the passive coupling. So this will be plus minus, and 1' to double prime coupling which is smaller, so therefore this will be like this, therefore this will be plus, minus. Okay. Now let us look at, this is the H1', now let us look at the H2'. H2' once again I will write 1', 2' is the active coupling. This is, what are the coupling constants here? So in this case I will have 2' to double prime, 2', 1', and 2', 3'. There are 3 couplings, okay, and the 2', 3' is not 0. So therefore let us start with this big one, 2' to double prime, that is the big one there. Let us say this is my, this is plus because this is the passive coupling, I am writing with the plus passive coupling there. And now what is this one? So this one is my 2' to double prime, and then I will have 2', 1'. Now this is my active coupling, okay, therefore this will be plus minus. This is the 2', 1', 2', 1', and 2', 3' is smaller than 2', 1'. So therefore this will be again another passive coupling. Okay. So now we see I will write here plus, and this is minus, this will be plus, and this will be minus. So what you got here? You got 8 of them, plus, minus, minus. So 8 components. Therefore this will have how many total in the structure? There will be 32 components, 4 into 8. So here I will, if suppose I want to write here, so this is plus, minus, minus. So and this side I have here plus, minus. So we put that here, so plus, minus, plus, minus, plus, minus, minus, plus, minus, minus, plus. So you will have 32 components, and all of these coupling, these will of course will have the separation there as we indicated here. So which is the, this is the coupling, this is the 1' to double prime, 1' to double prime. So by looking at these plus minus separations, you can figure out what are the coupling constants in each one of those. Okay. But often you may not be able to resolve the peaks in this manner. So what will happen? There will be cancellations in the middle, all pluses, minuses and things like that will cancel in the middle, then you may not be able to find all the peaks. So in such a situation what do? When we do not have this, of course you will have to calculate all of this. Similarly you can calculate the fine structures for the 2', 3' peaks, and then the 3', 4' peaks. You can calculate the fine structures for the individual sugar geometries. Okay. So when we do that, this is an experimental spectrum of the C2 prime endo sugar geometry, which is shown here. Okay. Now what you have here? So you have the 1 prime, which I already discussed with you. The 1 prime peak is present here, these are the diagonals here, and then you have the 2 prime to double prime cross peak present here. Okay, so let me write here, this is the 1 prime and this is the 3 prime, this is the 2 double prime and this is the 2 prime. Okay. So therefore you see 1 prime to 2 double prime and 2 prime, and then you see from this is the 3 prime. So 3 prime to 2 prime, notice 2 double prime 3 prime peak is absent. This is what I showed you earlier, 2 double prime, because that coupling constant is very small. And then you of course you have the 2 double prime 2 prime peak. Symmetrically you will also have these peaks here. Now these ones, as you can see, have a fine structure, these ones have a fine structure. This is what I was explained to you just now, and we will see how one can calculate these. Now you see, these are the simulations. So since there will be cancellations in the peak intensities, you cannot do it by simply, all the components are not visible to you, all the components are not visible to you. You have an experimental spectrum, you put in these numbers, certain numbers for the particular sugar geometry. Here what is calculated is for all the sugar geometries, starting from the pseudo rotation angle 9 degrees, 36 degrees, 72 degrees, 90 degrees, and this is my C2 prime endo geometry. And we instead of, see how many cross peaks are seen here, you only see 4 components, all the other there are lots of things which have merged, they have cancelled or merged and things like that. So in this particular case we see more components. If you see here, there is some cancellations here, some residual things are present here. So when you come to this area, then you do not see these cross peaks at all, because this is the 1 prime 2 prime peak. 1 prime 2 prime peak peaks around, the coupling constant peaks around 144. There is 0 here and 0 here, and therefore you will not see the cross peaks for the C2 prime endo geometry for these values of the pseudo rotation angle. Okay. Now therefore these are the patterns which are calculated and simulated, keeping in mind the resolutions that are there and the line weights. So once you have those, once you get these patterns. So experimentally what we have to do is, you will have to simulate and compare with the experimental spectra to estimate the correct values of the coupling constants. Okay. Now this is a similar simulation for the 1 prime 2 double prime peak. So this is the 1 prime 2 double prime peak here. Now you see there are many more peaks which are visible here, because this one has only 1 prime 2 prime coupling absent. 1 prime 2 prime coupling is absent, but all other couplings are there. See, because of the 1 prime 2 prime coupling is absent, you see this peak appears narrow along this axis. This is for the various values, because at this point at 9 degree the 1 prime 2 prime coupling is 0, right, for this and therefore at this value 1 prime 2 prime coupling is 0, therefore this side splitting is only 2, 1 prime 2 double prime coupling, therefore this is narrow. As we keep increasing, you start getting contributions from the 1 prime 2 prime coupling also, this actually becomes wider. You see, this width becomes more, this becomes more, further more, and it has the largest, see here is the largest width here, because the 1 prime 2 prime coupling is large and 1 prime 2 double prime coupling is also available here, therefore this has the larger width along this axis. There are 4 components along this axis that you can see that here clearly. Okay. In fact in this one you can also see all the 16 components, so to say, right. Centrally there are some cancellations at this point, okay, because of the plus minus, and you keep increasing the value of the P, go over to C2 prime endo geometry, and then you go over towards the end, once again it becomes narrow here. This is similar to this is again back into the north regime. North regime has a 1 prime 2 prime coupling is going to 0, so therefore this is becoming a narrower one here. So therefore from the looking at the patterns of the cross peaks in the cozy spectra, you can actually calculate, determine whether it is the geometry is in the north domain or the south domain. Okay. So I think we can stop here.